Okay, let's see. First of all, um, I thought I had put up the lecture from Wednesday. Is that correct? Has anybody seen the lecture from Wednesday up on YouTube? Has anybody tried to find it and not been able to? All right, well, I'll check it out. Okay. Um, so you guys are coming, so you're not using it. It's the people who aren't here that are using it. Anyway, okay. I inserted one slide this morning. This is a bit of a backtrack, something that I didn't explain. Well, I'm sure there are a lot of things I haven't explained well, but one that I know I didn't explain well. I just wanted to clarify it for you. So this is sort of backtracking just for a moment. We talked about the fact that different aspects of a visual stimulus are analyzed by different circuitry, which is in fact in different places in the brain, and in particular, location and color, or location and form, are analyzed in different places. This presents a problem, because suppose you have a picture like that, with a red thing here and a blue thing there, and suppose that location, i.e. this versus this, is analyzed in one place in the brain, and color, this versus that, is analyzed in a different place in the brain. How do you get the right color hooked up with the right location? And that is what's called the binding problem, and in fact it has not really been solved. So it's very, the previous slide that I used to illustrate this probably didn't make the point very well. That one. So this, I think, makes the point. Okay, now let's go on to where we were. If I can get the computer to move. Come on. God. Okay, actually, I think maybe this is where. Had I shown you these pictures yet? No. Okay, that's right where we are. Oh, come on. Something's going to get real slow. See what it does. Okay, we were talking about the fact that at the ganglion cell level, and that continues on further deeper into the nervous system, the ganglion cell level of the retina, and then on into further parts of the visual system, that one has this division into what's called color opponent cells, with the red-green opponency and the yellow-blue opponency. And that this arrangement kind of allows one to makes a number of predictions. So if these guys are inhibited by green and these guys are inhibited by red, then if you have a combined red and green, they're going to come to a point where they're going to balance out, and you're going to lose the sense of redness or greenness. You get sort of a colorlessness. Similarly, over here on this side. On the other hand, if you have this, if you have a red light and a yellow light together, that's fine. They're both represented. These guys fire and these guys fire, and you get a perception of orange. If these guys fire and these guys fire, you get a perception of turquoise or greenish blue, and so on, chartreuse purple. We're all familiar with those things. First of all, let me just show you this. Better. So you're familiar with the picture up on the top. Which is that this and this make yellow. And that's, if I balance the red and the green right, I would get white here instead. But I couldn't do that. My computer. However, if you balance, if you give a red light and a blue light together, you get purple. You're all familiar with that. But what you're not all familiar with, and which maybe is a little bit of a surprise, is if you balance a yellow light and a blue light, you get white. You lose the sense of color. And so it does just what that color square would say it should. But this raises some interesting questions to which there are not answers. One question is, So our subjective experience of color seems to correlate much, oh, I'm sorry, this is not really, it seems to correlate much better with the properties of color opponent cells than receptor cells. Thus it's these that we seem to see. So our minds, as it were, don't look directly at our receptors. It's as if our minds, our sense of consciousness, was looking at these guys. Our conscious perceptions behave as though they were looking at these guys. However, does this suggest that in order to have a sensory experience, we must have a particular kind of cell that fires when we have that experience, that is in some way responsible for the base of that experience? And that's a question we really don't know the answer to. In the context of vision, this color stuff specifically, what the question would really be is, okay, take orange here is reddish yellow. We all agree with that. Okay? But it's also, I mean, we see the color orange. It looks like a distinct color to us, and we give it a distinct name, orange. But ask anybody, and they'll say, yes, it's reddish yellow. Well, are those two different things, or are those the same thing? Is it possible that there are, in fact, orange sensitive cells that are selected for orange, and not just... Is what's happening when we perceive orange, in part, that there are actually special cells firing that represent orange, rather than just having red cells and yellow cells firing? Don't know the answer to that except a little bit, which I'll show you in just a minute. And when we look at this, suppose there really are orange, in quotes, orange cells, and we look at it and we say orange is reddish yellow, is that because there are, in addition to the orange cells firing, there are these red cells and these yellow cells firing? So the basic question is really only one question, and that is, if we have something that we experience as a unique quality, does that mean that there is some cell in the nervous system that responds selectively to that characteristic, that quality? And the answer to that really is not known. However, recordings from V4, which we've already talked about as being the color area, where a lot of color things are done, that's where color constancy seems to be computed, there is some evidence which suggests that there may be cells in that region that do go beyond red, green, yellow, and blue, and that there may be orange cells, etc. And this shows some of the cells that a fellow named Zecchi has found in V4. So in particular, here is a cell that, with the liberal interpretation, you might say was a turquoise cell. It peaks in the region that you can see as turquoise. Here is a chartreuse cell. Here is an orange cell. So maybe. But I also have to tell you that I've never talked to any visual neuroscientist who believed this data. Except for Zecchi himself, of course, who I've not talked to. This bottom line on this stuff is not in. But it's an interesting question. Okay. That's the basic stuff on vision that I want to talk about. We'll come back to making some general comments about it in a few minutes. But I want to turn now specifically to the issue of consciousness and conscious visual perception. And the neural correlates of conscious visual perception. I mean, to some extent, we've been talking about them. We're asking these questions about if we consciously perceive something as orange, does that mean there's a cell that corresponds to that? But I want to back up a little bit, or stand off to the side a bit, and look at this question of consciousness in a little bit more general way, but still using the visual system as the basis for the discussion, for the most part. Okay, first of all, consciousness is sort of the central fact of our psychological lives. If we weren't conscious, that's what it's all about. For many years, psychologists felt that whereas that was obviously true for each of us individually, that there was no way of studying consciousness because it wasn't an objective thing, it's only something that's inside each person's head. And so the belief was that this is just an issue that psychology can't address, can't think about, can't talk about. And so for well over 100 years, consciousness was simply not talked about in academic psychology. But fairly recently, within the last 10, 20 years, knowledge of the visual system has become sufficiently complete, and I'll have something further to say about the completeness of the knowledge in a little while. But knowledge of the visual, we've learned enough about vision that it seemed to a few visual scientists that maybe one could look at consciousness in the context of vision specifically. One of these people, actually, is Francis Crick, who fairly recently died, who's also, you all know from molecular biology and DNA. After he solved the problems of molecular biology, or practically created molecular biology along with Watson, he got interested in psychology, and in particular, one of the things he was interested in was this problem of visual consciousness. And then he collaborated with a guy named Kalk, who's at Caltech, who is still doing these sorts of things. In any case, first thing you need to realize is, and the thing which gives you an entree, which gives scientists an entree into studying what consciousness is and what it's good for, is the fact that all mental processing is not conscious. There's a great deal of mental processing that goes on that's not conscious. And we've already seen one example when we talked about the flexion reflex. If you step on a nail, you flex your leg, or whatever, you try to pull away from the thing that hurt you. But that happens, although it does hurt, you actually start to withdraw before you even feel that it happened. So this is and certain lesions of the nervous system can get rid of all the conscious aspects of it, and nevertheless the behavioral reaction goes on. So that is an example we've already seen of unconscious neural processing leading to behavior. A question that's interesting there, I'm not sure if I've raised this with you before, I might have already said this. The question that's interesting is, okay, if you don't need the pain in order to flex your leg, if that's not makes you flex your leg, why do you have the pain? And the answer to that is thought to be that the pain is a teacher. It teaches you to not do whatever it is you did that led to this situation on a future occasion, but it's actually not responsible for the withdrawal, for the flexion reflex itself. It's got another function. In the context of memory, there are also examples of conscious and unconscious processing. So suppose, for example, that I present you with maybe a couple hundred pictures of different people that you've never seen before, and you do it fairly rapidly, from one, dun, dun, dun. After you're finished with that, I show you some of these pictures together with some and I say to you, show you some pictures of people that I showed you the pictures of, and some new pictures that you haven't seen before. And I say, have you seen this before? And if I've shown you as many, hundreds of pictures, you'll have a very poor score. You won't remember very many. It'll get all mushed up. So you don't have a conscious recollection of having seen these, or at least you'll be able to remember some, but you won't remember very many. But suppose I do the experiment to evaluate whether you recognize these individuals in a quite different way. Suppose what I do is I flash each of these pictures very briefly with a bunch of noise around, visual noise, so that you're not even sure that there was anything there. And I simply ask you not, do you recognize this person, but do you see a face? It turns out that you're much, much, much more likely to see a face when I present one of the faces you've seen, than if I present one of the faces that you've not seen. And that is called visual priming. And it's basically an indication that even though you have no conscious memory of the thing, somewhere in your nervous system there obviously is a memory of this, otherwise there couldn't be this differentiation between the guys you've seen, the people you've seen, and the people you haven't. So that's an example of a conscious and one example of conscious and unconscious sorts of memory. Another example, which we'll talk about more next time, and maybe even at the end a little bit this time, is the distinction between what's called declarative and procedural memory. As we will discuss, you have two somewhat separate memory systems, or two different sets of memory circuitry in your brain. One kind of circuit, one kind of memory, is one in which your memories are conscious. So a good example is, again, in cases where you see individuals and you get a good look at them for a long time, you remember them. And so if you show a person a picture of somebody that they know, they'll say, yes, I recognize that person. Very easy. And there are lots of things, of course, that you recognize. That is an example of conscious or declarative memory. But there's another kind of memory, which is called procedural memory. For example, when you learn to play the piano, or learn to play even a piece on the piano, that goes into procedural memory. And if you ask somebody who does not have a piano in front of them, at least if you ask, maybe this is not true for a pianist, but if you ask an ordinary person, whether they remember how to play, sorry, let's do this a little bit differently. Suppose you took piano lessons as a kid, all right, and now 15 years have passed and you haven't touched a piano, and somebody comes up to you and says, can you still play Fur Elise, which is probably something you learned when you were taking piano lessons? You won't have a clue. The only way to find out is to sit down in front of the piano and see if you can do it, and you might or might not be able to. But the point is, this is an example, but you may be able to. There will be many cases where you can perfectly well do it, but you don't know whether you can do it because procedural knowledge is not accessible to conscious memory. In other words, it's memory that is shown only in performance and in doing something, not memory that you actually have a conscious recollection of. So that's another example of something where lots of stuff's going on, but it's unconscious. Another example of this in the context of vision is what's called blind sight. Suppose we have somebody with a region of ablation or damage in V1, primary visual cortex. If you get them to focus their eye on the middle of this page of print, they'll see, don't pay attention to the X for a moment, they'll see something that looks more like this. They'll see the print over here, but they'll see a blank space there. Suppose now that you present either an X or an O in this region of what's called scotoma in the vision, this absence of vision because of the damage of V1. They're not blind, their retina is okay, but their V1 is not. Present this X in the middle of this region of scotoma, this blind region. You present an X or an O, and you ask them, what do you see there in that region? And they say, I didn't see anything. They say, oh, come on, no, I didn't see anything, there's nothing there. Okay, all right, it's an X or an O. I don't know, I didn't see it. You have to choose, is it an X or an O? They usually choose X. Okay? So even though they think they can't see it, their nervous systems can see it and can process it and can make decisions based upon it. So that's a very famous, well-known, well-studied example of unconscious visual processing.
Is there an X actually drawn?
Yeah.
Okay.
Yeah, no, I mean, if they put an X, they'll see an X. If you put, I won't see. They'll, if forced to choose, they will choose the X. If it was an O, they'll choose the O. Now, this doesn't always work, but it's,
okay.
80% of the time or something like that. Yeah, no, yeah. Okay, so we have conscious and unconscious processing, and then that raises a bunch of questions. And it also, so we can study the problem of consciousness very well in the domain of vision because of this kind of phenomenology. So here's some questions that one might want to ask. One is, what does conscious as opposed to unconscious processing of visual information bias? What is the function of visual consciousness? If we can recognize objects and respond correctly, even though we say we can't see them, is it what's the difference? Is there something we can do when we are conscious of it that we can't do when we're not? Now, surprising as that may seem, nobody knows the answer to that question yet. Okay? It turns out there are a lot of technical difficulties to answering it, and the answer is not known. Secondly, what is the difference in the brain's representation of a visual stimulus that is consciously seen as opposed to one that is not? So, if we can be conscious, if we can process things both consciously, visual stimuli, both consciously and unconsciously, can we tell from the activity of the brain which ones are conscious and which ones are not? Which parts of the brain are involved in consciousness, etc., etc. Is there any way to, what is the neural signature of consciousness, conscious perception of and analysis of a stimulus versus unconscious? And then finally, why does a subjective sense of consciousness exist at all? And what really is it? Okay, question one is being researched, but there's not an answer. Question two is being researched, and there's some glimmers of answer. Question three, nobody has a clue how even to approach. All right, so let's talk about what we can talk about a little bit. Okay, first of all, we of course already know that different places in the brain are specialized for doing processing, which leads to conscious appreciation of different qualities. With movement over here, we talked about the moving disk, or color over here, but that doesn't really help us very much. That doesn't do very much for us by itself. But people have asked, what is the signature of consciousness? How can you tell whether something's conscious or not by looking at the activity of the brain? And there are two sorts of ideas floating around. One is that when something is conscious, there's something special about the activity. Maybe the firing of the nerve cells comes in some different pattern, maybe rhythmic bursts as opposed to single spike, it may, like as opposed to something like that. Or maybe when another possibility suggested is that there is a correlation in the firing pattern over certain different regions of the brain. So the activity, perhaps in the frontal cortex and the visual areas, occurs in a correlated way. So I can't render this, but basically, if there's a pattern of the firing, that pattern is correlated in time, or synchronous in time, maybe between the one area and another, and that sort of correlation is a correlate of being conscious. And there are people that think there's evidence for those things, and there are people who think that there's not good evidence for those things. Another way of thinking about it is maybe there is some special area that has to be activated by the stimulus in order for it to be conscious. And there are people who think that, and other people who, and other people who maybe don't think that. The two areas that have been most accused of being the relevant areas, if there are relevant areas, are the prefrontal cortex, which maybe isn't so surprising. That's the area where we supposedly plan, make decisions, and so on. So that's plausible. Or the parietal cortex. That's not quite so obvious. But the parietal cortex is part of the place that has to do with analyzing location and space. And there are parts of the parietal cortex that are especially seem to be involved in analyzing not just location in space, but also bodily, one's own personal location in space. And so if you start thinking about location and where you are, it has some flavor of the sort of thing that you might expect might be involved in a sense of being aware of something and being conscious of something. It's very loose reasoning, but anyway, let me show you some of the kinds of things that people see in the data, some data that people see that makes them think some of these things. This is a picture of drawings made by a patient with parietal lesions on one side. Now, this person can see bilaterally perfectly well, but each one of these pictures is only half the object. It's only what's going to one hemisphere and not the other hemisphere. This kind of data has led some people to think that the parietal cortex, so that the person sees it, but they apparently are not conscious of it. Or at least when you ask them to draw the thing, they just don't draw that half of the object as though they were not conscious of it. So this kind of data is consistent, doesn't prove, but it's consistent with the idea that maybe the parietal cortex has something to do with conscious visual perception. Now, yes?
Do they believe they've drawn the other side? I mean,
they probably, I don't know the answer to that. They probably, if you ask them, why didn't you draw the other side, my guess is they say, what other side? But I don't actually know.
So how do you know that they can see?
Oh, you can do very, like with our X and 0 test. So I'll have a test you can do to show that they see it perfectly well. I mean, by see, that they can respond, that their visual system can see it. Their consciousness doesn't see it, but their visual system does.
Does that mean that if you kept on bugging them about what other side?
Yeah, you would probably, might not even have to bug them very hard.
Okay.
Yeah. All right. Now, there is a technique which I actually don't think we've talked about in this course yet, called functional magnetic resonance imaging, but the newspapers enough that you probably all know about it, which allows you to measure the metabolic, the oxygen depth in great detail over the whole surface of the cortex and also some deeper parts of the brain. Since the oxygen debt is caused by neural activity, it gives some indication of neural activity. Now, exactly what aspect of neural activity is something that people don't fully understand, but it is viewed as a valid technique for finding out if a particular part of the brain is involved from some kind of information processing. And so the following experiment was done with this functional fMRI, functional magnetic resonance imaging, or fMRI. Moving grating was shown, but the contrast of the lines in the grating were made very, very weak, so that sometimes subjects said they saw it, and sometimes subjects said that they didn't see it. And then all this was being done in an fMRI scanner, and one looked to see what the pattern of activity was. And what was done is they looked at the pattern of activity when the subject said yes, and the pattern of activity when the subject said no, and they subtracted one from the other, so that you get a difference picture. And you can ask, well, did they, for increases or decreases between the two states, and these are drawn separately here. So there were increases on the yes trials in here, this is the side view, this is the top view of the brain, here, 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 here. So what regions are those? Exam. What? Sorry?
Parietal.
Okay, which one's parietal? That one or that one? That one, right, good. And here it is from the top view of it, parietal, yeah. And what region's that? No, it's on the cortex, both cortical. Sorry?
Occipital.
Yeah, occipital. Now, it's not very surprising you might get some occipital stuff, because after all, this is a visual stimulus. But it is interesting that you get different occipital stuff when the subject is conscious versus when he or she isn't. Yeah.
Is there a reason it isn't symmetrical? For the second image?
It was probably presented to one side. It was probably presented to just one side.
Okay.
I mean, I actually don't know the answer to that, but that's what I would suppose. Yeah. Okay. And then decreases. A little bit of sign here. I think that's, I don't know, I think it's parietal. I'm not sure quite what that is. But what's that? Frontal. Yeah, prefrontal probably. It's pretty far forward. Yeah, prefrontal. Okay, so those observations are consistent with both these ideas, that it might be parietal and it might be prefrontal. And so these are the kinds of data that people use to make the inference. And there's a lot of agreement that prefrontal and parietal are probably important, involved in the conscious aspects of visual perception. Yes?
What does the act of seeing something moving have to do with consciousness, though? Why did they link
It's because they can report it. Therefore, by definition, they're conscious of it. Okay?
or visually conscious?
Visually conscious. Conscious of they have the awareness of the stimulus. If we say, I would say, if somebody's aware of something, they're conscious of it. There's a long history in this field of, which has now, it's a situation that's now passed, but for a long time, people talked about consciousness in the context of wakefulness versus sleep. If you were awake, you were conscious. If you were asleep, you weren't. And there was lots of work on differences in the activity of the brain when you were awake and in your sleep. But that really didn't get at the issue. It was never very satisfying. This is more satisfying. But it's still just visual consciousness. Other kinds of consciousness, we don't know. This does not get at the question of, is the person conscious in the sense that they are alive and thinking? But it does get at the question of, are they consciously aware of something? Okay?
So these experiments can be done on humans, non-invasive?
These are human experiments.
Okay.
Yeah. Yeah, these are human. Another interesting observation is you can ask somebody to try to visualize in their mind some particular thing, and when you do that, everybody agrees that you get fMRI activity in the higher visual areas, like D4, protemporal cortex, so on and so forth. There's not agreement on whether you get activity in D1. Okay, so but that's still, this is relevant information. Now here is an experiment, an observation or an experiment that's extremely interesting. An observation, I guess I would say. This is a patient who has neurological problems. And this patient has two problems. One problem is they have a scotoma in their primary visual cortex, on the occipital lobe. So there's a black region there where they don't see anything. The other problem they have is they're epileptic. And epilepsy is commonly a temporal lobe problem. And what happens when they have epileptic seizures is you will often get an activity that starts at one end of the temporal lobe and just progresses along. Now, the temporal lobe has lots of memory circuitry in it. That's something we'll talk a little bit about later on. We're actually starting at the end of this hour, beginning of next time. And so, as this epileptic activity goes along here, this particular individual had visual imagery of somebody walking, as it turns out. Probably with some particular person walking. I don't know. Probably somebody they knew. And that in itself is kind of interesting. So as this progressed, they saw the person walking along. You can call it a hallucination, but it's acting on the right parts of the brain for doing something like this. So it's not. I mean, it's sort of it's a hallucination in the sense that there was nothing out there, but it's nothing. It's not their imagination. Their brain is really, the parts of the brain that should produce or might well be expected to produce some sort of image do, some sort of conscious image do. But what's interesting about this experiment is that as this thing moved along, and the person saw this individual walking along in their imaginary visual field, or in their mind, when this hallucination got to the part of their visual field where they had a scotoma in V1, the image vanished. And then when they came out the other side, and then it came out the other side. So what this suggests is that there may be some interaction between in order to have a conscious visual experience, there have to be some sort of interaction between V1 and, in this case, the temporal lobe, but V1 and some other part of the brain. I wouldn't say this experiment proves that, but it puts that kind of idea, at least into my head and into the head of the people that made these observations.
The hallucination of the motion of the man, or even the existence of the man?
Both.
Okay.
Just the whole experience.
All right.
Okay, now one comes to a very similar idea from a very different kind of experiment. One can't go around electrically. So, okay, the usual way of directly stimulating neurons in a brain for experimental purposes is to pass an electric current into the brain tissue in such a way as to depolarize neurons so they pass their critical firing levels and fire. But in most cases, you can't do that with humans, okay, for obvious reasons. But in recent years, a technique called magnetic stimulation has been developed, in which you pass a strong magnetic field to the brain, a moving magnetic field. A moving magnetic field, if you remember, you've had physics, you may know, causes an electric current to flow. And so you can cause a weak current in the brain by this magnetic stimulation. And whether rightly or wrongly, everybody agrees that this magnetic stimulation can't do any harm. I'm not sure the final word on that is in on that, but that's what most people believe. It's magnetic stimulation that you're getting when you get an MRI. Everybody thinks that's just swell. This is stronger than that. But anyway, so the point is these kinds of experiments are done, and everybody thinks they don't do any harm. Now, suppose we magnetically stimulate V1. And you don't stimulate all of V1, you do it in such a way that you're stimulating just a portion of V1. What happens is the subject will have an experience that's called a phosphene. Sort of an experience of light in some particular part of their visual field, flashing or something like that, sparkling, some sort of visual stimulation in some part of their visual field. And you can ask them where they see it, and this particular subject said, it's right around in here. And this is a normal subject. If you do the same thing, placing your magnetic stimulator over area MT, this area that's responsible for perception of motion, what happens is you again get visual stimulation, but now these are moving things that the subject experiences. So that's a normal subject. Now, if you do exactly the same experiment with a peripherally blind, somebody that's blind because there's something wrong with their eyes, you get essentially the same result. But what happens if you do the same thing with somebody who has a scotoma in V1? Well, if you do the stimulation of V1, of course, if you put the magnetic stimulator so that it's likely to be activating the neurons in the region where V1 isn't there anyway, or damaged anyway, nothing happens. That's not very surprising. That's not very interesting. But what is interesting is that if you stimulate area MT in the region that corresponds to the damaged area of V1, which is in the same part of the visual field, no perception of motion. So what that says is that the perception of motion depends both on some interaction between area MT, or what it suggests is that the experience of this moving visual stimulus depends upon some interaction between MT and V1. And even though it's MT you're stimulating, you might think when you're stimulating MT, the consciousness should see a moving thing. That doesn't work if MT is not connected up, or not, if MT doesn't have V1 there, the right parts of V1 there to function. So this again suggests some interaction between one region of the brain and primary visual cortex in the creation of a conscious perception of a visual stimulus. The same idea, and this is just sort of saying that with picture, so something like that seems like it might be going on in the perception of conscious perception of motion. Now, there's yet another kind of experiment which leads to the same sort of idea. This is an experiment on monkeys. So suppose you show a monkey a picture, a field, where almost everything is tilted in this upward left sort of, with an upward left sort of tilt, like so. But suppose that here there's a square in which the tilt is in the opposite direction. And you present that stimulus fairly briefly, so it flashes on. And the monkey has to indicate whether he saw the square or didn't see the square. He's trained to do that. That takes a while to train them up, but they can get good at it. But you flash it so briefly that sometimes the monkey sees it, and sometimes the monkey reports seeing it, i.e. is presumably conscious of it, and sometimes the monkey does not report seeing it, presumably is not conscious of seeing it. Suppose now that during this experiment you have electrodes, a recording electrode, implanted in V1 in a spot where we're sticking up a neuron that's responding to the portion of the visual field in this red dotted circle. And what you're doing, you make this monkey fixate on the center so that his eye stays in a fixed location relative to the stimulus. So we're just recording what a neuron that's looking at that little portion is seeing. You might think that what you would see is that when he says he sees it, that you wouldn't get when he says he sees it, you would get activity, and when he says he doesn't, you don't. Sorry, I didn't say one more thing I needed to say. This is a neuron that's responding in this region. It's also a neuron that is selective for this orientation of stimulus, because remember, V1 neurons are orientation selective. So this is a neuron which is designed to detect this sort of angle of orientation. You might think that when he sees the square, that you would get activity in the neuron, and when he doesn't see the square, you don't. But that's not what happens. It's different than that. What happens is, whether he sees it or not, you get the same initial pattern of activity when the stimulus flashes on. It doesn't make any difference. But what is different, depending upon whether it's not seen or it is seen, is that if it's not seen, what happens is when you flash on the stimulus, you get a brrr, which gradually tapers off over time in the neuron that's looking at this region. When the subject reports that he does see it, you get exactly that same kind of a burst, but then after about 100 milliseconds or something like that, you get a secondary burst. And that secondary burst comes at just the right amount of time for information to have gone from here probably to here and back again. So it's just the amount of time you would need for information to get from V1 to the next higher areas and then come back again. So the authors of this paper suggest that probably the consciously seeing the thing is correlated not just with activity in V1, but with this higher area also responding to the stimulus and sending information back to V1. So all these observations from these last 3 experiments that I've shown you are all kind of consistent in this idea that the development of a conscious visual perception depends on an interaction between V1 and other areas of the brain. And this experiment is the most informative in some ways, in strongly suggesting that it might be these, remember there are these backward projections from each area to each other area, and that maybe these backward projections are involved. And maybe it's when the backward projections are active that you get a conscious experience, which doesn't explain consciousness, but it's an interesting and useful observation. So the concept then is something like this. If here is V1 and here is, let us say, if here is V1 and here is, let us say, V2, V4, V3, something like that, some next higher area up. Suppose that this is a cell in this higher area that responds to, that has innervation from this area such that it will respond to a particular pattern down here. And suppose this is a cell, one cell that contributes to the fact that this pattern is recognized. So remember we talked about here in the projection from thalamus to V1 that the cells in V1 that respond to this orientation of line and this location in the visual field project back to excite those same cells. So at least at this level, which is the only place the details are understood, the back projection is closely related to the forward projection. And what happens here is that if you have a stimulus which is kind of meaningful to this cell, then this cell sends a message back that reinforces the neurons that contribute to that, to seeing that kind of stimulus. And the same thing might be going on at a higher level of the nervous system. So what this is saying is that when forward and backward pathways are both active, it means that something meaningful to the nervous system has been seen, because it means that the cell up here got a pattern of activity that made sense for its input connections. Those are in fact the ones that could be argued from these experiments that lead to a conscious perception of the stimulus. Well, if that's the way it is, that makes a great deal of sense. You want to be conscious of something that makes sense. If you get activity that doesn't make any sense, that's potentially just garbage, you don't particularly want to be conscious of it. And so this is a very plausible sort of way of looking at the thing, but nobody knows what it really means. But this is the sort of experimentation that's being done on this. Okay, so I want to make some conclusions about visual consciousness specifically and some other conclusions about vision in general. With regard to visual consciousness, at present the focus of research involves identifying exactly what are the properties of brain activity that occur when a visual stimulus reaches conscious awareness. One can anticipate that the next step will be to try to figure out what the consequences of various behavioral tasks, what the consequences are for various behavioral tasks of conscious versus unconscious appreciation of the stimulus. The final matter of understanding how the feeling of awareness arises is one that nobody's even beginning to think how to address. Okay, now let's have some conclusions for vision overall, not just visual consciousness. Okay, some overall take-home messages in the study of vision. 1. Our perceptions of the world are entirely constructions of our brains that are, in quotes, computed based upon evidence from our senses, our brain's inbuilt knowledge of the nature of the world, and memory. We've said that before, but it's very important. Secondly, the processing of sensory information is done by a bunch of separate modules, each specialized for analyzing particular aspects of the stimulus. How the analysis done by these separate modules is put back together and utilized in an integrated way, that is to say the binding problem, is not yet understood. Thirdly, how meaningful objects, for example, Jennifer Aniston, in the example that we saw, are coded is an important topic of ongoing research, since the neural representations of such objects must ultimately be used to make decisions, control actions, and so on. This is an extremely crucial question. the kinds of neural circuitry that would be needed for a distributed versus a dedicated code, which are the two kinds of ideas we talked about, would presumably be very different from one another. So resolving what the nature of the coding of meaningful objects is a very important thing, which is not fully resolved, although most people, as I told you, think it's a distributed code. The fact that, at least so far, nobody has found neurons whose activity seems to code for things in the center of a solid black square. Remember we talked about the fact that if you show a solid black square on a white background, it's only, in the earlier parts of the nervous system, or the visual system, it's only neurons representing the edges around the borders of the square that are active. The outside and the inside, there's very little neural activity, and yet we have a subjective impression that the inside is black and the outside is white. Very clear impression. People have looked hard, and nobody has found anywhere in the brain where neurons that you might expect would represent what's going on in the interior of that square are firing in a way that you would expect they would. And so that's what this is referring to. The fact that at least so far, nobody has found neurons whose activity seems to code for the center of a solid, things at the center of a solid black square. And there's the possibility that no such neurons exist poses a major challenge for understanding our perceptions, how our perceptions of the world are created. So that's a very interesting question. I mean, we'd be very comfortable if we saw somewhere in the brain there was something which was kind of like a bitmap of the stimulus, where basically, you know, all the neurons in the center of the representation of the square would be active, and those on the outside wouldn't, or vice versa. That would make sense. We'd be comfortable with that. It wouldn't explain things, but we'd be comfortable with it. But there's a very good chance that's not the way it is. There are a lot of people. Neurons have been looked for, have not been found, and there are people who are experts in this who think that they're not going to be found. And then that leads to a real problem for how you explain how our conscious, clear sense of that black square gets produced. Okay, from all this, at least to point, and all we've said, at least to point 5, visual circuitry is probably the best understood circuitry of the brain. Yet, it should be crystal clear from these lectures that there are huge gaps in our understanding of how it works. My guess would be that we know less than 1% of what there is to know. So there are people that you will talk to who will say, oh yeah, we really know a lot about it. We know so we can build a computer to do the same thing. Bull. Ain't going to happen. A lot more has to be known. We don't know. We know a lot, but we only know the very, very tip of the iceberg. And that's for the best understood part of the brain. Now, I don't mean to depress you. I'm taking this course. You'd like to think we know something to tell you, but that's, I think, the way that things are. Okay, I guess we've waited. I took too long to go into our next topic, so we'll start talking about learning next time.
Familiar with the race stems?
With what?
With the race stems?
Race tent? No.
Yeah, like pyrocetum.
I have no idea what you're talking about.
They're nootropics.
I still don't know what you're talking about.
They're like cognitive enhancers. Which,
okay.
They're supposed to be like safe, though.
Okay, what are they again?
Race stems.
Do you know what they're what they pharmacologically actually supposed to be?
No. No.
That's just you want me to answer you. I don't know yet. I'll try to find out. I don't know.
I mean, they're nootropics, but they're supposed to be safe. Like, they help like memory, working memory.
Okay, yeah, I don't know. They might be NMDA receptor helpers or something like that.
They are, yeah. How they work is
there are a bunch of things. Yeah, as neuroscience knowledge is getting, there's also a lot of stuff on affiliated bonding and the oxytocin. I don't know if you've heard about oxytocin. It's a hormone that's released in the brain that clearly has something to do with both forming parental bonds and forming sexual bonds and so on and so forth. And so people are selling a nasal spray, and it's supposed to, if you shine it, if you put it on at the right time, it'll make somebody else love you, or it'll make somebody else agree with you, or whatever. And there's little something to it. But probably not a lot.
Because I was at the cognitive enhancing discussion a while ago, where they talked about.
Yeah, I mean the point is these things are so crude.
Yeah.
Even if they work, they can't work very well.
Right.
Huh. But I'll have to find out about that. Yeah. Oh shoot, I meant to announce about that. okay, what's going to happen is my expectation is that next Friday we won't actually. I'll be finished lecturing and we'll have one here. But we also probably going to schedule an independent one Thursday late afternoon or early evening. But I'm not. I meant to ask our TA how we're going with that. I forgot. So actually, if you would on Monday, if you would raise that question with me before class, and yeah, good. Yes. Yeah. Oh, there you are.
Oh yeah. I'm gonna go talk to Elizabeth.
Yeah. So.
If you have a religious session, we'll be talking about something.
Oh no. I thought. Oh. I see. Well, that's a possibility. I was thinking late Thursday afternoon, but yeah, Sundays. I would. So I'd rather do it Saturday, but I guess people have other activities they would want to do on Saturday. In the other classes, what's typically been done, if anything?
I
PTA?
mean, the other class we always have it in the middle of the week, but I kind of have a weekend.
Before the exam.
Yeah, but I think it's been done. I just the classes that
I'd rather do it on Thursday. Yeah, I feel I don't want to. Yeah, I don't want to have it right before the exam. I'd rather people slack.
Okay.
And Saturday would be fine, but nobody's going on Saturday.
Okay.
You're probably not going on Saturday either. I'm not going. Yeah. So why don't you try it for late Thursday afternoon, 5 or something like 5 or 6, or very early Thursday evening, like 7 or 7:30, something like that. My own preference would be for the earlier rather than later. I'd rather do it and go home and have dinner and relax. And then to set it up. And then if we really are able to have a solid one on Friday, I'll be able to tell probably by next time how that's going to work out. We may decide we don't need it,
Okay.
but let's set it up.
Okay.
Great, sounds good. Yeah. Oh yeah, right. Yeah.
I was just wondering, when do you think the lecture for today and the one from Wednesday will be posted?